So hello, my name is Ethan Dionne. I'm also a member of Dr. Corneli's lab, along with Colby and Olivia. So I'll try and keep some of the context for my presentation short since they covered a lot of it already. But my project is actually one that I took up after we left PC and started our social distancing from home. So I actually did a bioinformatics project that involved measuring microscopy photos of Mycobacteriophage in hope that we could discover, morphological characteristics that could serve as a diagnostic for cluster assignment. So just to recap a little bit, phages are viral particles that infect bacterial hosts with Mycobacteriophage, specifically the genus of bacteria Mycobacterium. So these phages should be able to infect bacteria such as Mycobacterium tuberculosis and Mycobacterium abscessus, which are notoriously antibiotic resistant. So with the kind of work that Colby and Olivia are doing to genetically modify phages, we hope that phage therapy can be used as a technique to treat these kind of antibiotic resistant infections. So what I've done is I've measured phage particles, specifically siphoviral phage particles, to try and determine if there's a relationship between the genetic diversity of phages, which you can see in Figure 1. So Figure 1 shows an evolutionary relationship of phages and how they're designated into clusters. So clusters are kind of an arbitrary grouping of phages that are based on genetic similarity. So you can see the types of groupings in Figure 1 where they're marked with letters, and these letters designate these groupings of genetically similar phages. We also see in phage particles that there are morphological differences just by looking at microscopy photos. So you can see in Figure 2, these are some different Mycobacteriophage with siphoviridae morphology. And siphoviridae just means that they have long non-contractile tails. And then, of course, they have the characteristic capsid or head. So you can see in Figure 2 how there are many different kind of shapes, different tail lengths, different capsid shapes. Some of the capsids are more oblong, while others are more spherical. And then if you move up to Figure 3, there are also genetic relationships we know between these kind of different groupings of phages. So we have two different phages, Zolita and Skippy, which were two phages that were isolated on Providence College's campus in fall of 2018. And these diagrams show a genomic map of these two mycobacteriophages. This is just a small snippet to show a specific gene called the tape measure gene, which is actually found to be directly proportional to the length of the tail in the phage particle. So in this diagram, the tape measure gene is the longest green rectangle in each respective diagram for Skippy and Zolita. So with these kind of morphological relationships and these genetic relationships, we kind of hypothesized that there's probably some sort of relationship between the length of the tail and the diameter of the capsid and what kind of cluster designation the phage belongs to. So what I've done is I've gone through many, many microscopy photos of phages of different clusters, and I've taken these kind of measurements. And what I got was Figure 5 here, this graph. So what I did is I focused on phage clusters that are most common. Some of the most common phages that we've isolated in our lab come from these clusters. So cluster A1, B1, C1, E, and F1. And you can see that they kind of form these distinct kind of groupings and these nice kind of areas in the graph where they fell that show when you compare the capsid diameter and the tail length, there are these kind of distinct structural differences that you can sort them into different groups. So if we were to take a phage with an unknown sequence, so an unknown cluster designation, we don't know the cluster, and we were to measure the tail length and the capsid diameter, we could potentially kind of use this as a bit of evidence to assign it to a cluster without having to sequence it. This comes with one challenge, though, because not all of the phage clusters form these nice groupings on a graph. If you move your eyes to figure 6, I tested the K cluster phages, which Colby and Olivia talked about in their presentation. And we were interested in K cluster phages because they have a very broad host range. They're known to infect those notoriously antibiotic resistant tuberculosis and abscessus bacteria. So the K cluster phages didn't really land nicely in any of the groupings that I found with some of the other clusters. So unfortunately, the data isn't that clear cut, where we can just say, oh, all phages land into these nice groupings, and we can use this as, a pretty standard way to classify phages that are unsequenced into these clusters, because the K cluster phages don't share this kind of trend. So you might ask why, what use is this data to us? Well, it's very useful alongside different types of data. So in figure 7, I have a DNA restriction digest gel. And for those who don't know, a DNA restriction digest gel, you basically expose phage DNA, which you've extracted from the phage, to an enzyme that breaks the DNA at these certain sequences, which for our enzyme HA3 are common in phage. So what this does is it breaks different DNA from different phages into these kind of unique patterns in the gel that you can kind of differentiate between. So we can kind of determine that similar patterns must have similar DNA. And these similar DNA phages probably belong to the same cluster or the same subcluster. So I've kind of labeled this gel with to highlight some of these similar patterns in the gel with blue, orange, and then one labeled that corresponds pretty well with Skippy, who we sequenced as a B1 phage. So we can use this kind of technique and plot the microscopy photos on a graph after having measured the tail length and capsid diameter to kind of build evidence to make a cluster assignment, along with other techniques such as PCR as well. So in conclusion, I found that clusters A1, B1, C1, E, and F1 all have distinct structural characteristics when you compare tail length versus capsid diameter. And even though this trend doesn't hold true for the K cluster phages, we can use the data I've compiled in figure 5 along with DNA restriction digest data and PCR to make some conclusions about how to classify unsequenced phages, which is very useful to us because over the past couple of years, we've had freshmen in the fall have this kind of research experience where they get to isolate their own phage, and we have to choose 2 phages to be sequenced. And we want to discover new, unique, potentially useful phages and phage therapy on our campus, like we did with Zoe J in 2012. So this kind of data really is very promising to kind of improve our results with the freshmen to get more interesting phages on our campus. That's my project. Thank you.
Thank you so much. That's really interesting. Is there any relationship between the tail length and the other measurements you're taking with how many organisms that phage can infect? So what you're talking about, the K phages are more broad and the other ones are more narrow?
We don't have any evidence to suggest that the tail length has anything to do with the host range, but that would be something interesting to kind of investigate, along with a bunch of other factors, because these kind of things are very complex. There are different sites that phage can bind to on bacteria that are specific to certain clusters of phage. So unfortunately, we don't have an answer to that question.